Hello, welcome to Rocky by Chem series. In today's video, we will talk about registration mapping. Please, if you've not subscribed to my channel yet, kindly do so for me. Thank you very much as you are doing so. Now, registration enzymes recognize and then cut DNA at specific nucleotide sequences. So those regions that are recognized by the registration enzymes are called the registration sites. Some registration enzymes are such that when they recognize the registration site in the DNA, they are not going to cut the DNA through that particular site, but they are going to cut the DNA at some sequences that is away from the registration site. We classify such registration enzymes as type 1 registration enzymes. Type 1 registration enzymes do not cut the DNA at the registration site, but after recognizing the registration sites, they cut the DNA at some sequences that is away from the registration site. Now to the other class of registration enzymes, that is the type 2 registration enzymes, they cut the DNA along the registration site. They cut through the registration site. So we can define registration mapping as a technique that is used to map the location of specific sites within a DNA molecule that is recognized and then cut by registration enzymes. We are going to find out those regions in the DNA that are recognized by the registration enzymes and then cut by them. Mostly in genetic engineering, the type 2 registration enzymes are of interest because of the fact that they recognize the registration sites and then cut through them. So we can use registration mapping for a lot of purposes. We can use it for a lot of purposes. We can use it to do a lot of stuff like mapping the location of genes. Let's say we have a particular DNA and then we want to find out whether gene 1 comes before, let's say, gene 2. We can do registration mapping to find out that, and we can also use it to identify some genetic mutations. Some genetic mutations bring about a change in the registration site, and then as the registration site is changed, then it means that the registration enzyme cannot recognize such sites and then cut the DNA molecule. So when you are to run a gel electrophoresis, you will see such a DNA molecule with a mutation being a single band, but the one that is not mutated, we will be having two different bands due to the fact that the registration enzyme was able to cut through that one to generate individual fragments. And then we can also use registration mapping for the purpose of molecular cloning, that is cutting or taking some gene from one species and then introducing it into a vector. We can use registration mapping for that purpose. That is, through registration mapping, we can get to know the gene that we are looking for and then insert it into the vector. Now, what we are going to do is that we are going to find out how some calculations, or let's say how we perform registration mapping. Let's say we will be given a DNA, and then after giving the DNA will be subjected to digestion by registration enzymes, and then we will be asked to assemble the DNA fragments. So we will be using registration mapping to do the assembly of the various fragments generated at the end of the digestion. There is this small thing that I want you to be aware of before I proceed. There are two types of the digestion. We have single digestion and then the double digestion. For a single digestion, only a single registration enzyme is used. That is, when you use one registration enzyme to digest the DNA into various smaller fragments, we call it a single digestion. And then when two registration enzymes are used for the digestion, we call it the double digestion. So let me proceed. This is the question. A linear fragment of DNA is cleaved with the individual registration enzymes HIND3 and then SMA1. And with a combination of the two registration enzymes, the fragments obtained are, we have HIND3 to be equal to 2.5 kilobase and then 5.0 kilobase, and then SMA1 to be equal to 2.0 kilobase and then 5.5 kilobase, and then HIND3 and then SMA1, that is a double digestion, give us the fragment 2.5 kilobase per, 3.0 kilobase per, and then the 2.0 kilobase per. So we are asked to construct a registration map for this kind of digestion. The registration map is mostly constructed for the double digestion. we construct a restriction map for the double digestion. So let's start. I am going to divide this into various steps for things to be easier. So let's start with the step 1. For step 1, assign letters to the various fragments generated from the double digestion. Assign letters to the various fragments generated from the double digestion. We know the double digestion to give us 3 different fragments. That is fragment 2.5, 3.0, and then the 2.0. So we are going to assign letters to these fragments. So let's say letter A to be equal to the 2.5 fragment, letter B to be equal to the 3.0 fragment, and then the letter C to the 2.0 fragments. So that is what we are going to do under the step 1. So letter A for 2.5, letter B for 3.0, and then letter C for 2.0. Now let's move on to the step 2. For step 2, construct a table consisting of individual fragments from each of the single digests, and then find out which fragment from the double digestion will sum up to give us that fragment. We are going to construct a table, and this table is going to consist of what? The individual fragments generated from the single digestion. So for the table, one column will consist of the hind 3, and then the other column will also consist of the small 1 fragments. So we know hind 3 to be having 2 fragments, that is 2.5 kilobase pair, and then the 5.0 kilobase pair. And then the small 1 is also having fragment 2.0 kilobase pair, and then the 5.5 kilobase pair. I hope we have realized that both digestion will always sum up to the same length, that is 7.5. Both hind 1 digestion and then small 1 digestion will also give us 7.5. And when we consider the double digestion too, we are going to get the same 7.5 as the length. So we are going to assign the letters to the various fragments. So let's say from the double digestion, we said that let's make the 2.5 fragment to be equal to the letter A. So let's take the fragment 2.5 from the hind 1 digestion. We are having the 2.5 kilobase pair as one fragment, and luckily for us, we have the 2.5 kilobase pair fragment from the double digestion too. So this means that our 2.5 is going to be equal to what? Letter A. But in the case of the 5.0 kilobase fragment, we didn't generate that fragment from the double digestion, but we can sum up some of the fragments from the double digestion to generate this particular fragment. So from the double digestion, we have the 3.0 fragment and then the 2.0 fragment. So when you combine 3 and then the 2, you are going to get 5. So it means that the 5.0 kilobase fragment is equal to B plus C. Now let's move on to the digestion by the small 1. For small 1 digestion, we have 2 fragments, that is fragment 2.0 and then the fragment 5.5. For the fragment 2.0, we can straightforwardly get this fragment from the double digestion. So for the double digestion, we assign the letter C to the 2.0 fragment. So this means that 2.0 kilobase fragment from the small 1 can be equated to the letter C. And in the case of the 5.5 kilobase fragment, we couldn't generate this fragment from the double digestion, but luckily for us, we can combine 2 different fragments from the double digestion and then get this particular fragment that we are talking about. So when we combine the fragment A and then the fragment B, that is 2.5 plus 3.0, we are going to get 5.5. So it means that our fragment 5.5 kilobase pair is equal to A plus B. Now let's move on to talk about the step 3. For the step 3, we are going to arrange the various fragments in such a way that there are overlaps in them. So from the table that we can see on top here, that is A, the hind 1 digestion giving us the A fragment, the B plus C fragment, the C fragment from small 1, and then the A plus B fragment from the small 1. We are going to arrange these fragments in such a way that there are overlaps in them. So let me start. We are going to use one fragment to start the whole thing and then use it to find the other fragments. So we are going to start with the fragment 2.5, that is equal to the A. So let's look at this particular table. It is only the fragment 5.5 that gave us the letter A again. So we are going to place the letter A under the A here. We are going to place that fragment having the letter A under this particular fragment A. So 5.5 kilobase pair was equal to A plus B. So when we place the A plus B here, we are going to generate overlap. That is, A is going to what? Overlap with what? The A here. So the end of this particular fragment, that is the letter B, we are going to also place another fragment under it in such a way that we are going to generate another overlap. So for the 5.0 kilobase fragment, which was equal to B plus C, when we place that fragment here, that is the B here, will also be able to overlap with it. And then let's pick the last fragment equal to C, so that C will also be able to overlap with the pre-existing C that we have seen here. So after doing this, the next thing that we are going to do is to start the mapping itself. So for the mapping, we are going to draw our linear DNA. Though the question didn't state that we are working with linear DNA, but so far as the starting fragment is not equal to the ending fragment, it gives us a clue that we are working with a linear DNA. So we know from the double digestion that we got 3 different fragments. So we are going to divide the DNA, that is the linear DNA, into 3 different fragments. And then we are going to place the various fragments. We are going to place the letters here according to the way that they are overlapping. So A comes first, followed by B, and then followed by C. A comes first, followed by B, and then C, in the order that we are seeing here. And then the next thing that we are going to do is to place the various enzymes in such a way that the location that we are placing them is going to give us fragments that corresponds to the single digestion. So let's place the 3 enzymes in between the fragment A and then the fragment B. So when you read from the left side, we have only fragment A, which is equal to the 2.5. And then when you read towards the right side, we have the B plus C fragment, that is the 3.0 and then the 2.0, corresponding to the 5.5 kilobase per fragment. Anytime you are placing your enzyme, make sure that the fragments generated after the placement are always equal to the fragments generated by the single digestion. Now let's place the SMA1 here too. Let's place the SMA1 enzyme at this particular location too. So when you place SMA1 here and then read the fragments towards the left side, we are going to get the 2.5 and then the 3.0 fragments. And when you sum them up, we are going to get the 5.5 kilobase fragments that was obtained from the single digestion by the SMA1. And then to the right, we are going to have our 2.0 kilobase fragments. So this is all we have to do when it comes to registration mapping of a linear DNA molecule. I've given you a try work. You can try this particular work, and then when you finish, you can comment with your answer. And then I will review it for you. Thank you very much. Now let's continue. For the cases whereby we will be given a DNA that is circular. That is, some cases the DNA given will be circular. So let's see how to go about them. It is almost the same as the one for the linear DNA, but the difference is that the starting fragment is always equal to the ending fragment. The starting fragment is always equal to the ending fragment. So we are not going to do anything new. So let's consider this question. 2 registration enzymes, TAC1 and then ALU1, were used to digest a DNA, and then we are asked to construct a registration map for the digestion. So TAC1 alone gave us 3 different fragments, that is fragment 5.1, 5.4, and then 3.5. And then the ALU1 digestion also gave us fragment 6.5, 1.8, and then 5.7. Whereas double digestion by these 2 enzymes gave us 1.9 kilobase per fragment, 4.6, 0.8, 1, 3.2, and then 2.5 kilobase fragments. So we are asked to construct a registration mapping for this particular digestion. So let's start. The whole process is, as I said, divided into 3 steps. The first step is that let's assign letters to the various fragments from the double digestion. So for the double digestion, we obtain these fragments. So let's assign the letter A, B, C, D, E, and then F to them, as we did for the linear one. And then let's move on to the step 2. For the step 2, we are going to draw a table, and then for the table that we have drawn, we are going to pick the various fragments from the individual single digestions, and then try to find out which 2 fragments from the double digestion will sum up to give us each of the individual fragments from the single digestion. So starting from tag 1, we have the fragment 5.1. So for fragment 5.1, when you sum up the fragments A and then E from the double digestion, you are going to get your 5.1. That is 1.9 plus 3.2 is going to be equal to the 5.1. So our 5.1 fragments from the tag 1 single digestion is now equal to A plus E. Now for the 5.4 fragment 2, we are going to use B plus C because 4.6 and then 0.8 will sum up to give us 5.4. And then for the 3.52, when you sum up 1.0 and then the 2.5, you are also going to generate this particular fragment. You will also repeat this step for the ALU1 single fragments after the single digestion. So 6.5 will be equal to A plus B, 1.8 will be equal to C plus D, and lastly 5.6 is equal to E plus F. Now let's move on to talk about the remaining thing. We are going to arrange them, the fragments, in such a way that there are overlaps in them. We are going to arrange them in such a way that there are overlaps. So A plus E, we can just start anywhere. We can start anywhere since the DNA is circular. We can just start anywhere. So I prefer starting from A plus E. So we are going to place a fragment under it in such a way that you are going to generate overlap. So as we can see, the fragment 5.6 contains the letter E. So when you place it under it here, the E will overlap and then you're left with the F. So let's find another fragment which contains F so that the F here will also have its corresponding overlap. So we have the fragment 3.5 to be equal to D plus F. Take note that addition is commutative. So D plus F, we can just write it as what? F plus D. So that the F will be able to overlap with the pre-existing F. And then the D here too, we can use the 1.8 fragment having the C plus D. As I said, addition is commutative. So we can make the C plus D as what? D plus what? C. So that the D will also have its corresponding overlap. And then we will place the fragment 5.4. Here too, we are going to make it C plus B instead of B plus C. So that the C will also get its corresponding overlap. And then we are left with B. So let's look through and then find the fragment containing the B that we've not written so far. So let's use the fragment 5.4. So for the fragment 5.4, we have what? Okay, let's use the fragment 6.5. For the fragment 6.5, we have okay, let's use the fragment 5.4. From the fragment 5.4, we have B plus C. So when we revert it, it's going to be C plus B so that the letter C will overlap. And then the last thing that we are going to do is the fragment 6.5 having the B plus what? A. So that when we place them here, the B will also get its corresponding overlap. Now, the next thing is that let's move on to the circular DNA and then start constructing the map. So as we can see, the double digestion gave us 6 different fragments. So for the 6 different fragments, let's divide the DNA into 6 different fragments. Let's divide the circular DNA into 6 different fragments. And after dividing it into the 6 different fragments, we are going to place the enzymes here. We are going to place them according to the way the fragments are assembled. So as we see, A is the one that we are starting with. And then after A, E follows, followed by F, followed by D, followed by C, and lastly followed by B. So as I said, we place the enzymes based on the way they did their digestion. So as we can see, for the tag 1, I place it in between the letter B and then A because when you combine the fragment 1.9 and then the 3.2, it corresponds to the 5.1, which is found in the tag 1 single digestion. That is why tag 1 enzyme is placed here. So you place it at the beginning of the A fragments and then at the end of the E fragments. And also we have the fragment 2.5 and then the 1.5. We have the fragment 2.5 here, as well as the fragment 1.0. That is when you combine these two fragments, you're going to get 3.5. And then 3.5 is also found in the tag 1 digestion. And lastly, we are going to get our 2.2 kilobase and then 4.6 kilobase here to also be to be a single digestion by our TAC1 enzyme. Yes, so that is why the TAC1 enzyme was placed at this particular vantage points, and then the remaining points will be for the ALU1. That is how everything is going to be. Yes, so thank you very much for watching this video. Please don't forget to subscribe.